Hi, everybody. Now that we've talked about protein synthesis, let's talk about what happens when something goes wrong. So, with DNA, there's a bunch of steps that happen here. So, DNA replicates itself during mitosis when it needs to make a new copy. DNA is constantly being opened to read the instructions, the genes, to make mRNA that code for the proteins. And in these processes, on occasion, mutations can happen. So in the replication process or because of environmental factors, it might cause changes in the DNA. So let's take a look at what a mutation is. So mutation is just basically any change in the DNA. So remember, our sequence is important to tell us the instructions to how to make the proteins for each gene. So different factors, either a mutagen or a carcinogen, can cause a change in that sequence of DNA. Things like radiation or ultraviolet rays or different chemicals you might be exposed to. So a mutagen is just in general any substance that can cause a mutation. And a carcinogen is something that causes a mutation that leads to a cell becoming cancerous. So like ultraviolet light is a carcinogen that can alter changes in your DNA in your skin cells that can cause skin cancer. The chemicals in cigarette smoke can cause changes in the DNA in your lungs that can lead to lung cancer or tumors growing in your lungs because it alters the DNA so that it doesn't work properly or it does not control cell growth, and that's how we get tumors. We talked about that a little ways back when we talked about cancer. So there's three different forms of mutations. And our first mutation is what's called an addition, which is where a base is added. So if this was one side of our DNA strand, what's going to happen in an addition is we're going to add an extra base in that strand. So now instead of having 9 bases in this segment, or maybe this is a gene, now there's 10. Okay, and what's going to happen is when we take our normal gene, so we've got our normal gene and it codes it into, this would be our mRNA, and this would be broken down into our codons, right, in groups of 3. And then we would look at our chart to figure out which amino acids these codons code for. So we've got pro, glu, cis, and gly. That would be a normal whatever protein this is. And this will fold up in a specific shape. And that's what, proteins are one of the key things with proteins like enzymes and antibodies is that their shape is important. So if we did an addition to this same normal gene, so maybe through some kind of mutagen causes an extra base in there. Now we've got a kind of a floater base here, and everything kind of gets shifted over one. And so our first codon would be the same, but our second codon is now CGA as opposed to being GAG. So this is going to code for a different amino acid, and you can see every amino acid after that first one is different than the original. So this protein is going to fold in a different manner or in a different way, and it's going to be not function the way it's supposed to because it's going to have a different shape. Okay, so that's what happens with an addition. We're adding an extra one, and all the bases get shifted down one. Our second one is a deletion, which is just what it is. We're deleting or removing one of the bases. So again, here's our original strand. It would be as if we were removing one of these G's. So there's 3 G's here. We've lost one somehow, whether that's whatever the cause of the mutation is. And so again, now we've got our code, our normal genes gives this as our amino acid sequence for our protein. If we delete one, so in this case we deleted this T here, and so now our second codon, it was GAG, now it's GGG. Okay, And so what's also going to happen is you'll notice that um, we would end up with one less um, amino acid as well, because here this would be 1, 2, 3, 4, 5 codons, and now there's 1, 2, 3, 4. The fifth codon is missing one, so we actually wouldn't have this val unless we knew what the rest of the sequence was. So again, it switched to all of the amino acids, and so this is going to make a different protein with a different shape. Okay, so our third mutation is called a substitution. So we are switching one base for another. So again, if this was my original strand, in some process, whether it's during replication or it's a mutation because of a carcinogen or something like that, one of the bases is going to be switched in for a different base. So instead of G, we have a C here. And so what happens with this, it usually only affects one of the codons, but that one codon might affect the sequence of amino acids. So again, here's our normal, we get our sequence. With the substitution, here we've switched out this second T for an A. So in our codons, we now have a U, so it goes GUG instead of GAG. And if you look that up on your codon chart, that actually gives you ARG instead of GLU. So you've got one different amino acid, and believe it or not, that one amino acid could affect the entire protein and how it folds. An example of this in kind of a real life example is something called sickle cell anemia. So with that, our red blood cells are normally this kind of round shape, and inside it is the protein hemoglobin that has this kind of shape normally. And so these nice round red blood cells can fit through the blood vessels, and they can just flow freely. Someone that has sickle cell anemia, their gene sequence actually changes just one base, which changes one amino acid, and it causes the hemoglobin to become clumped like this. And because that hemoglobin that's making up the red blood cell actually has this kind of sickle shape. It's not nice and round. And so somebody that has sickle cell anemia, some of their blood cells are like this, but then they also have some that are the right shape because they would receive two different copies of this gene, one from each parent. So maybe one parent gives them the gene that causes this shape, and then the other one causes that shape. So these ones that are sickle shaped, the problem comes is that these these red blood cells can become clot and block off blood vessels. And that's just because of one base is different in the sequence of the DNA. One last thing about mutations. Mutations can be passed on from parent to offspring only if the mutation is in the gametes, so in the sperm or the egg. So like if you get a mutation in like a skin cell, so like if I were to get skin cancer, I wouldn't pass that on to my offspring because that mutation in the gene is only in my skin cells and the skin cells that form from that one cell, not in the eggs. In order for it to be passed on, it has to happen in the gametes. Same thing with like lung cancer or something like that. Those are specific mutations to specific parts of the body, and so those don't get passed on. It only happens when it is in the sperm or egg. And so that is all there is about mutations. I hope this helps with your understanding, and I'll talk to you next time.